Hello, I'm Natalie Pierce,
And I'm Lana Abdul Mohsen.
and this is our project on macroevolutionary patterns in the Tyrannidae family. So our goals of this presentation are going to be, we are going to be giving some background on the Tyrannidae family. We are also going to be talking about a work of literature on the genus Enierates, which is a part of the family. We will be talking about a study found in that work of literature on that genus. We'll be talking about our hypothesis, be showing a phylogeny that we reconstructed, the conclusion we drew from that phylogeny, and we'll be comparing our tree to the tree from the literature. So a little bit of background on the species. So the family of the species is known as the Tyrannidae family, with the common name the tyrant flycatcher. It is the largest known family of birds, with about 438 species in total. Now, this makes it great for scientific study, and even to this day, scientists are still attempting to reconstruct phylogenies of relationships of species to each other, as shown in the paper we did on the genus Enierates. It's still a question on how all these birds are related. So some distinguishing factors of each species include things such as the color of the bird, the tail length, the beak shape, as well as body shape, whether the bird is more round or elongated. The birds also are found in South and North America. However, the three regions we are looking at for our reconstructed phylogeny are Mexico, Bolivia, and Brazil. So a work from the literature that we did was on the genus Enierates, and this study focused on 14 out of the 17 species in the genus. Now, the tree that they created was not monophyletic. In fact, they noted in their study themselves that the group was paraphyletic. So the phylogeny they created outlines relationship based on geographic location and climate. So those found in similar geographic and dry regions are much closer related to each other than they are to those found in humid regions. On top of that, the different colored leaves indicate different taxonomic groups. So the study from the literature, it focused on reconstructing the Enierates phylogeny based on geographical location as well as climate. This was doing a reconstruction of a previously made phylogeny, and it divides the genus into two regions based on temperature, either being humid or dry. It found that those in drier regions are much closer related to each other, whereas those in humid regions form their own group. It also created a sister clade called Euromeus for those in a humid region, which resurrected the idea of this sister clade existing, as previous phylogenies and scientists had said that it was not a thing.
So, our hypothesis is evaluating the species of Tyrannidae, and it is looking at whether they are more closely related by country or by ecomorph. For our reconstructed phylogeny, we focused on the CYTB gene. This gene is useful when determining phylogenetic relationships, and it was also used within the literature paper that we mentioned previously. It is used because of its sequence variability, and it is very good at determining relationships within families and genera. We used GenBank to obtain this data, and for our methodology, we used birds within the same family, and we used birds within the same order. We also used the three geographical locations that we mentioned earlier to select these birds. So we picked the birds based off of their location in Brazil, Mexico, and Bolivia. We also selected the birds based off of three colors, green, orange, and brown, or mixed with brown, as some birds were green and brown or orange and brown, and we wanted to include them within the same group. It is important to note that for our hypothesis, we would expect to see a monophyletic group surrounding the geography, or a monophyletic group that surrounds the ecomorph, which would refute our hypothesis. And as we see from our reconstructed phylogeny, the leaves are the colors of the birds. So a green leaf indicates that the bird is green itself, and the brown leaf indicates that the bird is brown, and a orange leaf indicates that the bird is orange. We decided for our outgroup that we would use a species that is also a bird. It is from the same order, but it is a different family, and we wanted to do this because we didn't want our tree to look too distant and separated. Like initially, we had used a reptile, and the tree itself looked too separated. For the branches on the phylogeny, we decided to use the branches as the geography, and so the red indicates that the bird is from Bolivia, the green branch indicates that it's from Brazil, and the light blue branch indicates that it's from Mexico. The numbers on the branches indicate the bootstrap value, and that's basically the branch support value. So a value of less than 0.7 indicates that the branch is not well supported, and a value that is greater than 0.9 indicates that the branch is very, very well supported. For our conclusion, we found that our phylogeny actually refutes our hypothesis, but it is neither forming a monophyletic group around geography or ecomorph, but we do see that it is closely related by ecomorph than by geography, where we can see that birds that are green form a smaller paraphyletic group, and birds that are brown form an even smaller paraphyletic group, and similarly for birds that are orange. So we can see that there is the similarity between the birds of the same color, that they are more closely related. For the comparison of the trees, we noticed that from the literature that we compared our tree to, we both evaluated geography, and the difference was that we evaluated based off of the ecomorph color in addition to geography, whereas they evaluated it based off of geography and the climate within that geography only. We noted that both of our results concluded that the phylogeny has paraphyletic groups rather than monophyletic groups. An important difference is also that we decided to root our tree with one outgroup, whereas they rooted their tree with multiple outgroups. Additionally, we evaluated our hypothesis based off of the species within the family, whereas the Tyrannidae family, whereas they evaluated it based off of the genus within the Tyrannidae family, so theirs was more specific and more focused on a genus.